So we have a little more material to get through in our antiviral section, and then we'll switch gears and we'll start talking a little bit more about the immune system and start focusing on anti-inflammatory type compounds and how they're mediating their effects, especially in disease states such as arthritis and asthma. So now I want to focus on anti-HIV agents and other antiviral agents for the types of viruses that actually integrate into the chromosome. And so these infections are much longer lived within the host and they're harder to deal with because when the DNA from a virus actually integrates into the chromosomal DNA of the host or the infected person in this case, it actually hides and it evades from any of the immune response that's going on within the person's body. So immunity is really a condition where antibodies are being created and they're released into the general circulation. And so they go around and they try to target foreign substances that are inside the bloodstream or inside the body, but not inside a particular cell. So we don't typically have antibodies on the insides of our cells unless they're being produced by our immune system, and then they get expelled into or secreted into the body fluids. So if you have a virus that actually integrates into the host chromosome, it can hide there for a very long time. And a lot of viruses will actually do this and integrate. The HIV virus is a very good example of a virus that will integrate into the chromosome. Some of the other types of viruses that integrate into the chromosome would be the herpes virus. So that's another very long-lived infection that you typically cannot clear. The chickenpox virus, also another one that will incorporate into the genome. And so people that have had chickenpox can have flare-ups with it later in life that actually cause the condition of shingles. So what happens when people have flare-ups, such as like the herpes outbreaks too, when they have an eruption of a cold sore, for example, from the type of herpes virus that would infect the mouth area, what happens is that under stressful conditions, there are signals that will activate the virus when it's in its hidden state inside the chromosome that will cause it to start replicating itself and go back into a lytic state where it's actively dividing and using the host machinery to create new viral particles. And so it's like you have these bursts where you have an additional viral infection that's going on within the host. And that happens with the HIV virus as well. And so antiretroviral drugs have been designed to try to inhibit the lytic phases of the virus. So we can't totally clear out HIV infections. So once a person's infected with the HIV virus, they're going to have it for their lifetime. We don't have any way to get it out of their chromosomal DNA once it's been integrated into the host. However, we've got a lot of effective agents now that help really treat these flare-ups or outbreaks of the lytic phase of the virus where it starts taking over the host system to make new viral particles. And so these antiretroviral agents would include nucleoside analogs. And so one of the first steps for these viruses that require reverse transcription from an RNA molecule to a DNA molecule, or even some of the viruses that require their own DNA polymerase to enter the cell and be utilized to initially replicate like a single-stranded DNA molecule as well, before it integrates into the chromosome, these drug molecules can be very useful at blocking this activity. So this would be a specific enzyme that is a component of the viral genome that is not represented in the host, and so we can develop very specific or selective agents that recognize the polymerases that are used for either reverse transcription or for the DNA polymerase that might be of viral origin. So these would be the first two classes, our nucleoside analogs and our non-nucleoside reverse transcriptase inhibitors would be of this class of compounds. So they're targeting the actual replication of the viral genetic material. We also have a lot of compounds that are actually protease inhibitors as well. And we'll see for the HIV virus in particular that a number of the proteins that are used to make both the capsid and the actual coat proteins that are on the envelope, they have to be actually cleaved or cut after they are transcribed and translated into the protein sequence before they become a mature protein and can be utilized to actually build the capsule of the virus around the genetic material. And so if we have protease inhibitors that actually block the processing and cleavage of the proteins, of the viral proteins, then they can block the maturation of these protein molecules, and they will be non-functional in building the scaffold that would be required to create the capsid and the nuclear envelope for the virus. And so you never can get a full viral particle that can actually bud from the infected cell. And so typically for people that have HIV, they have a combination of at least 2 antiretroviral agents that will help block the activity of the lytic phase of the virus. And so having this cocktail therapy where you have multiple drug agents that you're actually taking, this will enhance the potency, and it also helps delay this process of evolutionary resistance to our drug compounds or our therapy that we're trying to treat the patients with. So here's a general overview of the HIV life cycle. It's not much different from the hepatitis B virus that we saw, from the hepatitis viral example that we saw before, except that this virus is a positive stranded RNA virus. And so how this works is the genetic material is housed, again, inside this capsid. So the capsid is surrounding and protecting the genetic material. The HIV virus also has an envelope, and so this is made up again of a lipid bilayer, and it's got specific proteins that are expressed on its surface. So these proteins, actually some of them, the GP120 protein in particular, recognizes a protein binding partner that's called CD4 on the surface of T cell lymphocytes. And so these are one of our major antibody producing cells within the immune system. And this is the target cell that gets infected by the HIV virus. So when it binds to the CD4 receptor, it's going to be able to fuse with the plasma membrane. This allows the entry of the capsid into the cell. And so you have a pre-integration complex that forms. It's going to open up the capsid, so the capsid starts breaking down, and what you do is you release the genetic material in here and the enzymes that are bound in an inactive state within the capsid. Once they're inside the cytosol, there's enough of the other materials that are required to make these enzymes active that you immediately start getting the reverse transcription of the viral RNA into a DNA molecule. Once you have a double-stranded DNA molecule built, this will actually integrate into the host DNA. If it sits in the integrated state for a long period of time, this would be the latent phase of the virus. So remember that latent just means hidden or occult. And so this means that it's going to stay hidden and not cause an infection during this time that it's integrated. So there's signals within this viral genomic sequence that will sense different stress levels to this cell. And so if the viral genome senses that the cell is under some kind of stress, be it exposure to some toxin or actual stress that the person might be under, like at work or something like that, this can cause the virus to actually move from the latent phase into the lytic phase. When it moves into the lytic phase, it actually starts the transcription of the viral genome. So now you're starting to make new viral RNA. This RNA is going to serve as the genetic material that's going to get housed in the new viral particles that are going to get formed. In addition, you also have the translation of the viral RNA in this case, because it's positive stranded that gets packaged, and it's also the positive strand that you can use with the host ribosome to actually make viral proteins. So all of these viral proteins get made, and some of these viral proteins include proteases. And the proteases are actually going to help make the mature form of both the coat protein, so you've got the envelope proteins that are out here, and you also have the capsid proteins that are going to help serve and form this nice barrier that actually will house the genetic material in the mature virion. And so these proteases are very important for the maturation of the protein products, cleaving them into the right shape so that you can get this very nice crystalline structure that surrounds our genetic material for the HIV virus. And so when this happens, you can see that the formation of the virus would be complete. It's in a mature phase, and it's going to actually bud off of the host cell. And so you can have hundreds or thousands of viral particles that would actually bud off of the cell until you mediate the full destruction of the host cell. So the virus just goes into this lytic phase, and it basically uses up all the resources in the cell until there's nothing of the cell left. The cell is just destroyed. You use up all the plasma membrane, and the cell just keeps shrinking until it finally bursts open and breaks. So what happens then in the HIV infection is that you have this annihilation on the T4 lymphocytes, which are very important for mounting normal immune response within the infected person. And so the person becomes very immune compromised when you have the HIV virus go into the lytic phase, because the T4 lymphocytes, which are producing a huge portion of the antibodies for this person to protect them against all types of infections, are now dying. So, and when they die, then it takes away the person's ability to fight off other infections such as pneumonia, things like cancer. Kaposi sarcoma are another common complication caused by the immune deficiency that occurs with a full-blown AIDS case. And so a lot of antiviral drugs then have been targeted at these different features within the HIV pathway to help try to prevent the infection and the spread of the HIV virus, and to keep it, once it is infected, in a quiescent or latent stage, so that it's just integrated in the chromosome, but that it never comes out and shows or rears its ugly head. And so many of the targets for this have been the reverse transcriptase process. So this process of generating DNA molecule from the RNA molecule is a novel process for this virus. The host cell does not have this particular enzyme, and so this reverse transcriptase is a perfect target for drug molecules to be developed to. And we'll see the bulk of our antiviral agents actually target this process right here. Some of the other viral medications target this process over here during the lytic phase. They actually will target the maturation of the major proteins that are used in the formation of both the capsid and the nuclear envelope. And so it basically inhibits this maturation process of the viral particles and blocks them from forming full units of virus, and it blocks them from being able to be emitted from the host cell. And so it protects the host cell overall from infection and from cell death. So there's a number of different HIV subtypes, and the one that causes the most human infections is called HIV-1. And within the HIV-1 group, you can see that there's a diversity of different types as well. And the M group, in fact, is the one that is the most virulent strain. And so we should say one more thing about the HIV life cycle. When you have this process of transcription in the reverse direction, the enzyme that mediates this process of converting the RNA back into a DNA molecule is not highly efficient, and it makes a lot of mistakes during this process. So the viral genome is constantly being mutated into just a little bit different form every time it gets replicated and integrates into the host genome. And what this does for the virus is that it makes it very prone to evolution. And so drug compounds can become resistant, and so this virus can become resistant to drug compounds very, very quickly, because it has a very, very high mutation rate that's going to allow it to evolve much more quickly than many other viruses. And in fact, within a single person, they may have 300 different strain variants within their body based on the HIV infection and mutation rate. So this is a very awful feature of the HIV virus that has made treatment more difficult in coming up with agents that can retain their usefulness within the clinic. And the prominence of blocking this evolutionary strategy has been using a cocktail approach to treatment. Treating with multiple agents at different phases in the pathway will help inhibit or block this evolutionary process by being able to catch multiplying virus at multiple stages in this pathway. So we said with the subtypes of HIV, HIV-1 is the most common and the group M causes the most infections in human. There is an HIV-2 virus. It seems to be less virulent, and it's more of the class that is going to actually infect monkey species instead, although the HIV-2 class is known to cause AIDS in humans as well. So, but it seems to be a less virulent form. So by comparing sequences between these two viruses again, we may be able to learn more about why the class M organisms are so much more virulent in human systems. So, but for now we're going to talk a little bit more about the types of compounds that are out there to help treat the HIV virus or the HIV infection. And so there's three major classes of drugs that form up the cocktail treatment for an HIV positive patient. These guys are nucleotide reverse transcriptase inhibitors. So they actually mimic nucleotides that would be entering into the growing nascent change. The bases, the G, the A, the T, and the C, that normally get incorporated into the growing DNA molecule as it's being converted from RNA into DNA. These guys would mimic those structures and get placed inside the growing DNA molecule as it's being made, and it actually will block the synthesis, and it would cause faulty synthesis to occur. So you would actually be blocking this step here. You wouldn't be able to form the DNA molecule. You have non-nucleotide reverse transcriptase inhibitors as well. And what these guys do, these guys actually bind specifically to the enzyme, and they're going to inhibit the enzyme activity. So they don't look like the bases or the substrate that would go in there, but they're still going to bind and block the activity of the enzyme. So our other major class is going to actually be our protease inhibitors, and these guys are going to be involved in splicing our polyproteins into their mature or their final structures, and these guys again are going to be utilized to make the protein capsid that surrounds the genetic material, and it's also going to be utilized to make protein core structures that will show up in the envelope of the virus as well. And so this blocks the maturation of the virus, and it inhibits the release of the virus from the host cell. And so it blocks the release, and this will actually ensure that the host cell will survive and still maintain its normal function within the body. So you can see that there's other areas in this pathway that can also be targeted for drug development. These guys just have not been as easy to exploit in this process of drug development, but they do still serve as targets, especially this HIV integrase. So there's an enzyme that is required, that is one of the viral enzymes, that's required to be able to splice or incorporate the viral genome into the host chromosome. And so this integrase enzyme would be another great target for drug development, in that the host system does not have this type of integrase enzyme present within their own bodies. So this is an example of some of our nucleoside reverse transcriptase inhibitor, and what you see is they look a lot like a DNA base. So if I just look up the DNA bases really quick. Okay, so this is a nice diagram that shows the different nucleotide bases. Actually, these are the nucleoside bases. So only the sugar molecule is attached right now. So you see the ribose sugar down here. And these would all be for the RNA. The deoxyribose would be lacking the OH at this position. And you can see that our molecules over here actually are mimicking these structures. So instead of incorporating the normal DNA bases into the growing DNA molecule that's being reverse transcribed by the HIV virus, they'll incorporate these alternative fragments here, and these guys don't contain the 3' hydroxyl group, and you won't get extension of your product after this. And so DNA replication can no longer occur if you incorporate one of these alternative bases into the growing chain. So these have been very effective agents in blocking the reverse transcription process of the HIV virus from going from RNA into the DNA molecule. So this should prevent integration of the virus into the chromosome. So further infections within the host patient should be blocked using these nucleoside reverse transcriptase inhibitors. So their mechanism of action is twofold. They offer competitive inhibition of the reverse transcriptase enzyme, and they also can be incorporated into the growing viral DNA chain and cause termination of the replication process. These drugs do require intracytoplasmic activation. They need to be phosphorylated first into the triphosphate form. So our nucleotides are not just integrated into the DNA molecule as the nucleoside product. They need to actually have the phosphate incorporated as well. So if we look at one of our bases for DNA incorporation, this one is adenine. You can see the nucleoside just contains a side of sugar, but the nucleotide also contains the phosphate group. So these guys have to be activated into the phosphate form so that they can be incorporated into the growing DNA molecule. And they actually need to be the triphosphate form to be incorporated. So they need to have 3 phosphate groups extended here. When they get incorporated, 2 are cleaved off during the reaction. They release the energy that's required to link this to the next nucleotide in the growing chain. So that's why they require activation within the cytoplasm. Typically when things contain phosphate groups, it makes them very, very polar and much larger molecules, and they typically cannot be moved around within the body very easily if they're phosphorylated. So pre-phosphorylated drug compounds don't work very well. You have to wait until you get them inside the target location, and then they have to be modified or activated into the phosphorylated state typically to mediate their action via this route. But these drugs do have good activity, and they do get phosphorylated into the active component, and most have activity against both forms of HIV, the HIV 1 and 2. So this is just showing another diagram of our HIV virus and the major targets of our drug molecules. So we've been talking about our reverse transcriptase inhibitor blocking the pathway for the formation of the genomic RNA into the double-stranded DNA molecule, and then integration into the host chromosomal DNA. So reverse transcriptase inhibitors are very popular. Integrase inhibitors have been less developed. Protease inhibitors are also very popular. And then you can also try fusion entry inhibitors as well that would block this interaction between the GP20 protein of the HIV virus and the CD4 molecule on the surface of the cell. So these are just some additional nucleotide analogs. The AZT, or azithromycin, is a nucleotide reverse transcriptase inhibitor, and so it's one that's shown up on this page, and we'll just use this one as an example for how these guys are mediating their activity. So it's well absorbed from the gut. You can see it's a pretty polar molecule, so it's going to be very soluble in water. The phosphate groups tend to trap the compound on the inside of the cell. They become too big to move across the membrane through big channels if they have those phosphates incorporated. So these molecules move from the gut to the body tissues and fluids, including cerebrospinal fluid. And they're eliminated predominantly by renal excretion following metabolism in the liver. So they have glucuronic acid that would get incorporated into the molecule, help make it more soluble so that it could be excreted in the urine. So these types of drugs, these nucleotide reverse transcriptase inhibitors, will decrease the rate of the clinical disease progression, and they prolong survival times for patients that are infected with HIV. They're also effective in trying to treat HIV-associated dementia and thrombocytopenia, and this is partly because they can cross the blood-brain barrier and they can get into the cerebrospinal fluid. They can also reduce the rate of vertical transmission of HIV, so if mothers are infected with HIV, it's not necessarily transmitted then to their daughter or their son because these guys can help block the transfer of the virus through the placenta. They do have some adverse effects though. They can have myelosuppression. And so what is myelin? Myelin is the special cells that actually surround the axons of the neurons. And so we'll briefly just take a look at an axon. So if we take a look at a common neuron or a brain cell, you can see that these cells have a lot of different parts associated with them. The large extension here is called the axon, and this is the one that's going to send nerve pulses from this cell to its downstream target cells so that it can maintain its function within the pathway to relay signals from the brain to the rest of the body. The incoming signals are taken in at the dendrites from local regions near the neuron cell body. You can see the axon can be very, very long. It can extend all the way from the end of the spinal cord at the base of the spine all the way down to the big toe, because all of the cell bodies are contained within the central nervous system, so within the brain or the spinal cord. So these axons can sometimes be very, very long. And when they send their signals all the way down the axon to the neighboring cell, some of the signal is going to dissipate or be lost out the sides of the neurons in the axons. And so these Schwann cells, or oligodendrocytes in the central nervous system, will actually wrap themselves around the neuron like a little blanket. And they wrap themselves around the axon, and they inhibit the loss of the signal all the way down this axon. And so what happens in patients that are being treated by these nucleotide reverse transcriptase inhibitors is that some of the side effects of these drugs is that they show cytotoxicity to the cells that actually create these myelin sheaths that are going to surround and protect or insulate the axon. And so degradation of the myelin around the axon in the neuron causes some loss of feeling or neuropathy that can occur in these patients. It can also disturb motor control too if it affects the motor neurons and the myelin sheaths that are surrounding those guys as well. You can also have anemia or neutropenia, so it can affect the function of red blood cells and cause anemia, or the white blood cells and cause neutropenia as well. This can lead to some inhibition of immune function from the treatment, and so these guys can be more susceptible to other infections, or have weakness or fatigue fairly easy from the anemia. You can also have some gastrointestinal intolerance, headaches, or insomnia. So while we become more effective at treating HIV so that we can get it into a state where it doesn't cause the death of the patient, the treatment with all of this cocktail of HIV inhibitors is pretty rough on the body. There's a lot of side effects that go along with these drugs. So preventing AIDS spread and AIDS infection is really critical. So there's some other viral DNA polymerase inhibitors, and so these guys would infect the DNA polymerase enzymes that are specific to viruses like the herpes virus. They also can affect and inhibit the HIV reverse transcriptase as well. And so these acyclic nucleotide inhibitors are also mimicking the structure of the DNA bases or the nucleotides. And so you can see they have names like acyclovir. You may have heard of this one, or valacyclovir. And what they do is that instead of having a sugar molecule down there, they just have an acyclic chain here, or a straight chain, that can mimic that structure. And this actually will competitively inhibit the DNA polymerase because it still will bind with the DNA polymerase and cause problems. So our herpes virus is a double-stranded DNA virus. This is largely affected by these acyclic nucleotide compounds. So the viral infection would occur, and then you have a viral DNA polymerase enzyme that's involved in the initial replication of the DNA molecule from the virus. And so you get replication, and this replicated form of the DNA will then get integrated into the nucleus of the cell, where it will integrate into the host chromosome. So our nucleotide inhibitors, the acyclic compounds, would enter the cell. They get phosphorylated again, and they can get incorporated then into the growing chain, and it will block the extension step because there's no sugar molecule to extend the next nucleotide on. So the nucleotide blocks replication of new viral DNA or RNA molecules. So this is our acyclovir compound again. It gets activated to the phosphorylated form, So the phosphates get linked on here. It can get incorporated into the extending chain. And what happens is it's going to block the further extension because there's no place to put the next residue onto. So these guys are really active against the DNA viruses especially. And so they're active against the herpes simplex virus, type 1 and type 2. They're active against the varicella zoster virus, which causes chickenpox. And they're also active against the Epstein-Barr virus and the cytomegalovirus, which can cause complications, including forms of cancer with the Epstein-Barr virus. So this is a very nice set of compounds that's very active with DNA type of viruses. So this is just an example of inhibition of HIV DNA elongation. And so this is with our acyclovir is mimicking our guanosine residue. And you can see that we have inhibition of our reverse transcriptase in this case that's mediating the elongation of the DNA molecule off of the RNA template. So unfortunately, when these agents are given as a single agent to the patient, they can develop pretty high resistance pretty quickly. And this will result in cross resistance to other nucleotide inhibitors too, such as the valacyclovir, the famciclovir, and the ganciclovir. And so usually the activity of these drug compounds gets inhibited because the enzyme that mediates the phosphorylation or activation of the pathway to activate the drug compounds in vivo gets altered, and so you don't get the phosphate groups put on there. And without the phosphates, they can no longer be incorporated into the growing chain and actually cause chain termination events to occur within the cell. So some of the clinical uses for these types of drugs, they're used in the treatment of herpes simplex virus infections. This would be the first selection for our acyclic nucleotide inhibitors. The chickenpox virus, the varicella zoster virus, is less susceptible to acyclovir than the herpes simplex virus. And so higher doses are required to help inhibit this virus, especially when it comes out again, causing the shingles symptoms seen in later life. Some of the adverse reactions of the acyclic nucleotide inhibitors would be nausea, diarrhea, and headache. So they have fewer side effects than the reverse transcriptase inhibitors that we talked about a few minutes ago. And some of these derivative compounds, like the valacyclovir, have been mediated to try to reduce the resistance levels to some of the other acyclic drugs. So some of these other modified versions of the drug compounds have been modified so that they can have quicker availability and mediate their responses much more quickly within the body. So this valacyclovir is an extended unit that contains a valine amino acid that gets extended onto the acyclic component, and this guy gets activated like 5 times faster than the acyclovir does and can get incorporated that much quicker into nucleotide extension, and so it's a little bit more effective. So it has higher potency than the acyclovir, but they both can reach the same efficacy in their treatment or inhibition of the DNA polymerase. This one just gets there faster. And then ganciclovir is another cyclic derivative. And this one is more specific against the cytomegalovirus and has much better efficiency in this case against the cytomegalovirus. So it has 100 times greater efficiency than the acyclovir does. And then you also see myelosuppression and also neutropenia as side effects using these acyclic derivatives as well. And then there's some other analogs that are also used for treatment of viral infections against either the reverse transcriptase or the DNA polymerase. So there's also some non-nucleoside reverse transcriptase inhibitors, and these include things like telaviridine, nevirapine, and efavirenz. And so these guys are binding directly to the reverse transcriptase enzyme, and they're actually inhibiting it. So they bind near to, but they bind at a distinct binding site of the nucleotide reverse transcriptase inhibitors. So they bind to the enzyme at a unique site away from where the nucleotide reverse transcriptase actually binds to the enzyme. So these are just some of the compounds that are utilized as non-nucleoside reverse transcriptase inhibitors. And these guys would be used then as part of the highly active antiretroviral therapy called HART, and this is traditionally used for the treatment of HIV virus. And so this guy is utilized in this combination therapy, or this cocktail that would be utilized to help block the activity. So a typical HIV patient might be taking a nucleoside reverse transcriptase inhibitor, a non-nucleoside reverse transcriptase inhibitor, so double targeting this pathway, and then they also might be taking a protease inhibitor at the same time. So these have specific activity against HIV. The NNRTIs were developed specifically for treatment of HIV virus. And there can be cross-resistance among this class of reagents that develops within the HIV itself. And so as they develop resistance to one of these agents, typically they're going to have mutations that occur in the reverse transcriptase enzyme that also make the enzyme not bind to the other non-nucleotide types of reverse transcriptase inhibitors. So if you get resistance to one, you usually generate resistance to the other drug molecules in this class as well. So in the major adverse reactions for these guys are skin rash that's associated with them. The next class of inhibitors are the protease inhibitors. So again, these guys are going to be required to actually cleave the proteins that are used in producing the mature viral particle, and so they're utilized in creating the capsid proteins, and they're utilized in also creating the final proteins that will be incorporated into the viral envelope. And so by inhibiting the maturation of these viral proteins, you actually inhibit the formation of the full viral molecule as well. And so you inhibit budding and release of the viral particles from the infected cell. So this is just an example of a protease inhibitor, the saquinavir. It was the first protease inhibitor that was marketed for treatment of HIV. And so what you see in this slide is the infected cells and the release of the viral particles into the bloodstream. And so this drug is actually going to block this budding process. So again, combination therapy with multiple agents that target different areas of the HIV virus, of the HIV viral life cycle is very important. Again, because having the reverse transcriptase enzyme going from RNA to DNA is a very ineffective process that causes lots of mutations to the HIV molecule as it is transferred from an RNA molecule to a DNA molecule. And what this serves to do then is give the HIV virus this potential to really evolve quickly. It has lots of mutations very, very quickly, and it can become resistant to different drug treatments very, very quickly as well. And this is bad for the HIV patient because then they would go on to develop full-blown AIDS, which is a horrible disease to have to die and suffer from. So by taking these combination therapies that are going to hit the virus in multiple places of its life path all at the same time, it's much less likely to be able to develop multiple evolutionary strategies all at one time to overcome the force of the antiviral medications that are being taken if you're taking multiple antivirals at the same time. So the cocktail approach for HIV treatment is very, very critical, and patient compliance with their medications is also very critical for maintaining the actual function of the antiviral medications. So these patients have to be very careful that they keep taking their medications. So these combination therapies, though, do come with some adverse effects. We've already talked about the neuropathy effects, and so deadening of specific neurons, especially sensory neurons, is a common phenotype that is seen in HIV-positive patients that are on this cocktail therapy. They can also have syndromes that alter body fat distribution, and they can get what's called a buffalo hump or truncal obesity around their midsection. And they can also see facial and peripheral atrophy of the other parts of their body. They can also have insulin resistance, and so they can start developing diabetes in association with their treatment for HIV, and they can also develop this condition of hyperlipidemia. And so this is hyperfat formation, and so they will start forming and storing fat molecules, especially around the truncal location, so around their midsection, and that mediates this truncal obesity that's seen within this condition. So those are the main agents that are used as the cocktail agents to fight HIV. And then we also saw some other nucleotide inhibitors that were used for DNA viruses as well, such as the herpes simplex virus and the virus that causes chickenpox. So there's also some immune enhancing agents that are also utilized to help combat or treat viral infections. And so their mechanism of action, they're going to actually compete for the receptors on the surface of the host cells so that you can block infection or docking of the virus onto the host cell, and you can avoid absorption or penetration of the virus into the host. And so this would uncoat the viral nucleic acid possibly and cause the genetic material to be destroyed before it actually infects the cell, where it can either cause the lytic phase of the virus, or in the case of viruses that integrate, would cause the latent stage of the virus. So these agents could also block viral biosynthesis, and they also can enhance the host immune activity. And so we'll talk about a couple of agents that do this last effect here, including the interferon group of compounds. So the interferons are actually endogenous proteins. So endogenous just means that they're made inside the host normally, and they exert complex antiviral and immunoregulatory functions within the cell. They also have anti-proliferative activities as well, and so these can be preventative proteins that would help protect against the formation of cancers as well. So they mediate their activities through altering cellular metabolic processes, and so they can be involved in regulating gene expression through the synthesis of both the RNA and the protein molecules. And so these are not actually specific antiviral agents, but they actually function to cause elaboration of effector proteins in the infected cells. And so they can result in the inhibition of viral penetration or uncoating. And so this is kind of a secondary reaction that's caused by these interferon proteins. And they can do this by altering the viral transcription or creating viral messenger RNAs, or they can block the virus from being translated once the messenger RNA has already been synthesized. Or they may also interfere with virion assembly and release. So the interferons are actually a family of proteins, and they are classified based on their cell types from which they're derived, and they have some slightly modified gene sequences that encode for these proteins. So interferon alpha, beta, and gamma are the three major homologs of the interferons, and they fall into two major type classes, type 1 and type 2. Type 1 are found in leukocytes and fibroblasts, and type 2 are found in other immune cells. So these known enzymes are induced by interferons, and so interferons can mediate their function by activating a protein kinase that leads to phosphorylation of elongation factor 2, and so this can result in inhibition of chain initiation of peptide chain initiation during translation process. So it's going to alter proteins that are involved in protein translation. It can also alter proteins that lead to the degradation of the viral messenger RNA before it can be incorporated into the viral particle that's being formed, or before it can be utilized for translation processes to create viral proteins. So you can block protein synthesis, you can block or cause degradation of the transcriptional process or the messenger RNA, and phosphodiesterases can actually degrade the terminal nucleotides of tRNA molecules and help inhibit peptide elongation as well. And so the interferons appear to mediate these functions by altering gene expression patterns. And so if you look, they activate common signaling pathways. So we've been talking a lot about receptors. The interferons, alpha and gamma, are going to bind to their subsequent receptors, and they're going to activate downstream pathways that will activate transcription factors that bind in a sequence-specific manner and will alter the expression of downstream target genes that are involved in maintaining an antiviral state. So they've been useful in the treatment of things like chronic hepatitis or AIDS-associated Kaposi sarcoma and other forms of cancer treatment, as well as well. They do have some side effects and toxicities such as neutropenia and anemia, so they're affecting the bloodstream very, very quickly, killing off both red blood cells and white blood cells. So this can cause flu-like symptoms, including fever, chills, headache, myalgias, and other fatigue symptoms. So this is just an example of another immune-enhancing agent that's used. And you can see this looks like a lot like a double-stranded DNA molecule, or a portion of a double-stranded DNA molecule. And so what this does is it actually is an inducer of the interferon system. And so this causes immune enhancement again due to the generation of the interferons that are produced from this subsequent pathway. However, there is high allergic reaction to this compound. So it's a very big compound that would be given. It's also very polar and has these phosphate groups on it that don't enter or get absorbed very well. So what's our conclusions for our viral section? If we have further understanding of our viral genomes and the viral receptors, antiviral drugs will be more effective. So the more we keep learning about how viruses work, the more effective we're going to be able to make new drug compounds. In collaboration with this, we also need to have an understanding of how cells react when they get infected with viruses as well. So understanding these processes, like the interferon process, can also lead to whole new classes of antiviral drugs if we can have further immune boosting systems that can cause our own immune system to become more effective at recognizing viral infections and dealing with the viral infections as they're happening. So overall, antiviral drugs are still pretty new to our system of treatments and system of drug compounds. And they're still pretty experimental right now. So hopefully there will be further discoveries and new processes of synthesis to be discovered for the generation of new viral compounds. So I think we will stop there for this section.